Okay. Yeah. Yeah, it's quite exciting how high your viewership numbers are going, and also the success of your current books. Yeah, that's wonderfully exciting. And
Well, we'll see. I mean, yeah, the popular book on quantum medicine, I think it's an experiment. We'll see how it sells, and
yeah,
I hope it will.
I expect it to do very, very well. And anyway,
I may
and I can help
I may
make
be risking my academic reputation, but I don't.
no nonsense. No, I think it's a good thing for you to be doing. Yeah.
Great, thank you.
So let's just see here. Yeah, so I think we can proceed. We have 3 students, and we probably will get more. So let's take it away. Yeah, quantum biology part 4.
Thank you, Kim, and thank you everybody for sticking around for the final installment. And today I think we'll hit a few notes on the quantum biology, quantum medicine theme. I'm starting with kind of a historical comment on somewhat related concepts of coherence, which are the coherence and synchronization, which are sort of part and parcel of quantum behavior, but they originated a long time ago by people like Alexander Gurevich, and the Soviet Union, who measured some electromagnetic fields around living systems. And so not all electromagnetism must be quantum, but could be, and especially could be coherent. So so that led to the kind of monumental work of Herbert Froehlich, who is shown here on the left. He towards the end of his life, he switched from solid state physics to biophysics, and he proposed a theory which would explain the emergence of coherence in biological systems, and that was based on interactions between, and it was a microscopic theory related to cellular membranes, and was based on interactions between phonons. We talked about phonons earlier as quanta of mechanical oscillations in condensed matter systems, and in addition to it, excitons, which are electronic excitations. And in this particular case, he was invoking membrane phospholipid groups in membranes of living systems, and hypothesized that they can be oscillating in synchrony, giving rise to a quantum coherent state. And that was kind of revolutionary at the time, 1970s, and was based on several assumptions, one of which was the structural organization of the phospholipids in the membrane, their dipole moments interacting strongly, and then realistically adding the physiological temperature as kind of a noisy environment. And on top of that, which was kind of critical, and I need to spend a minute on this, energy pumping. So as I think I mentioned a few times before, biological systems are distinguished from inanimate matter by metabolism, and that means that they are in a state of far from equilibrium, far from thermodynamic equilibrium, and this is what Froehlich took advantage of, and postulated that some unspecified form of energy is pumping these modes and taking them to a so-called metastable state, not the ground state, the lowest energy state, which is the state of equilibrium, but that's a state of death. But living systems would create an oscillating dynamical state, which would be characterized by metastability. Now, there were many attempts at proving this, and even today, I think there are papers occasionally, one or two a year, which demonstrate some way of this fairly condensed state being present in proteins or cells, but it has not been really embraced by the scientific community after decades of trying. But I don't think it's a flawed theory. It was just not properly positioned. And what really struck me recently when I thought about this, because I wrote a few papers about this topic, that the most natural place where this kind of behavior would possibly take place is neurons, where you actually have action potentials delivering electrical energy quasi-periodically, but more or less constantly in neurons. And to my knowledge, this has never been applied to neurons, and that's kind of bizarre, but I'm actually working on it now. So now this brings me to the next topic of sort of quantum or quantum-like phenomena in biology, and that's the concept of the intelligent cell, which was proposed by Günther Albrecht Bühler from Northwestern University in Chicago, and he has a fantastic website. You just Google intelligent cell, and you'll find it hundreds of pages of illustrations and movies from his laboratory. And he basically performed experiments whereby he tried to demonstrate that cells interact with each other or communicate with each other electromagnetically, not chemically, not by physical contact, but by electromagnetic interactions at a distance. And that is one major step toward quantum biology, because these would be photons, and this is a diagram here showing you sensitivity with the range of wavelengths which overlap between visible and infrared ranges, and so that's kind of intriguing. The two questions that emerge are, first of all, where do biological systems produce photons? And that's actually a big area of biophotonics that I have no time to even scratch the surface of, but suffice it to say that there are several possibilities, one of which is the byproduct of mitochondrial function, so once again, energy production. And another possibility would be by reactive oxygen species recombining and emitting a photon as a result of this recombination. So that's a mission, and clearly in both scenarios related to metabolism. And now the question of transmission, and of course that's by electromagnetic waves, and then reception. And here's a big kind of open question, which structures in the cell would be sensitive enough to receive a photon and transmit the energy in some form for mechanical transduction, because in the end you have movement, motility towards the other cell. And so Albrecht Bühler pointed to the centriole, which is shown here. It's an organelle, a very important organelle, the central enigma of cell biology, composed of 9 triplets of microtubules, forming this symmetrical cylinder-like structure, and a central cartwheel, I think it's a doublet of microtubules. So again, microtubules, which I will highlight later on, play a prominent role here. And he argued that these centrioles, their architecture is such that it allows for very accurate orientational determination of the source of photons to within a few degrees in both in-plane and out-of-plane directions. Now, so I'll leave it at this because it's a longer story, but very interesting and worth spending some time on. Another somewhat related topic to it is the so-called bystander effect, which is quite well documented in the literature, and it shows cell-cell communication, whereby one of the cells, for example, in a tumor, is hit by high-energy ionizing radiation, and suffers some damage and it signals to the neighbors that there is some danger or potential damage coming, and they actually react without being directly hit. They react to it by overexpressing, let's say, heat shock proteins or some other defense mechanisms. And so that's kind of an interesting topic of how they communicate related to Albert Buehler cell intelligence. This actually has a larger dimension as well. And there is a researcher at McMaster University, Professor Carmen Madrocil, who's been studying this at the level of organisms, including various species of fish. The experiments were conducted really, I highly recommend reading these papers that were published out of this research group that indicated, in fact, inter-animal signaling without any chemical or hormonal signaling or physical touch. They did experiments with some fish schools in aquaria, and one group was exposed to radiation and the other was not. And the one that was, both groups were expressing the same, overexpressing the same proteins, which indicates some kind of communication at a distance, let's put it this way. Even more bizarrely, in one of the papers, they reported that the response of the fish occurred before they were actually exposed to radiation, which really speaks to the issue of quantum entanglement, possibly, or such exotic effects. This is, of course, no proof, but it introduces a real possibility of quantum effects, these exotic quantum effects taking place in animals and perhaps even humans. So we'll come to it. The question, of course, is how do these signals get amplified from the cellular level to the tissue, to the organ, and to the organism? I don't think there is an explanation, but it's definitely worth exploring. So that's the future of medicine for you. Perhaps even effects like that affect human beings. I'm not familiar with an extension to the human population. These are some key papers, recent papers from the group. So that involves the biophotons, and of course that's quanta of electromagnetic radiation. Now I will switch gears now to another topic that is under the banner of quantum biology, and that's quantum metabolism. And again, I don't need to re-emphasize the importance of metabolism as a crucial characteristic of living systems. So this has a fairly long history, going back to 1780, in fact. The energy consumption, the so-called basal metabolic rate, as it scales with the size of organisms, and that was studied by these famous scientists, Laplace and Lavoisier, in the late 1700s. And then Rubner in 1904 started scaling these relationships, and finally Kleiber in 1940 made a systematic study and determined mathematical relationship between the size of an organism and the metabolic consumption rates. Interestingly and counterintuitively, it's not doesn't scale in proportion to the size. You would expect that if somebody, not somebody, but let's say if you compare an animal which is 10 times bigger than the other, it would be requiring 10 times as much food intake, calories. No, it's not. In fact, this is a so-called allometric scaling relationship, where W is the body size and Y is the metabolic rate. And you have two coefficients, alpha, which is kind of species or organism specific, and beta is an exponent, which is characteristic of all biological systems by and large, with some exceptions. And this beta, that's the scaling relationship, is 3/4 typically, not 1, which would be the case of linear relationship. There are some slight variations and differences. For example, plants, the exponent varies a bit between 2/3 and 1, but for animals is by and large uniformly 3/4. This has, of course, various practical implications. For example, the doses of drugs, medication is not linear, is not in proportion to the body mass, but it scales. Typically, I think the doctors, one of the doctors, I don't know if you prescribe medication, but would scale it with the surface area, body surface area, which actually is 2/3, because surface scales with the square of the distance and volume with the third power. So it's 2/3, close enough to 3/4, but it should strictly be 3/4 scaling relationship for dosage. Now, why am I talking about this in connection with quantum biology? Because this problem has been solved after decades and decades of head scratching and different attempts, and because different organisms have various ways of producing energy from bacterial membranes to chloroplasts in plants to mitochondria in animal cells. And so Lloyd Demetrius, a colleague of mine, professor at Harvard and Max Planck Institute, he came up with this idea that observed mathematical similarity between the scaling relationship in allometric scaling laws and the relationship between the specific heat of solids and temperature, as I alluded to in, I guess, my first or second lecture when we talked about quantum physics, solid state physics, crystals and lattice vibrations. So I will not go into too much math, but the derivation of this relationship by Lloyd Demetrius is very elegant, and basically postulates that metabolic enzymes act as oscillators. They have a typical characteristic turnover time, and not only that, but they are coupled or synchronized across the cell, and each cell has about 2,000 mitochondria or so, and maybe 50 ATPases. So it's about 100,000 enzymes working in concert across the cell, and of course, billions and billions across the human body. We are all kind of as an organism synchronized metabolically. So that makes a lot of sense. And when you take this into account, this quantization of metabolic oscillators along the lines of the Debye theory, it all comes together. But before I show you the bit of math that may be a bit sophisticated, I want to also mention again that there is solid evidence for quantum tunneling of electrons and the electron transfer chain of mitochondrial inner membranes. So there is already the quantization in strict sense taking place. So Lloyd Demetrius postulated that these metabolic oscillators and ATPases basically have characteristic time or frequency, and then introduced the Planck-like quantization formula: energy equals a constant times the frequency of oscillations. So that's the first analogy, and the second was this Debye-like postulate that these are wave-like phenomena. So we have phonons in crystals. Here we have waves of metabolic transitions. Okay, so these are the assumptions of the theory of quantum metabolism. Enzymes undergo electrochemical oscillations. Oscillations are generated by metabolic energy supply and associated with the transfer of electrons. And and then there is the quantization principle. And here is the formula, which is reminiscent of Planck's formula, which started quantum physics in 1900. This is going back to 2003, almost 100 years later, or just over 100 years later. Planck-like quantization of metabolic oscillations in living systems. Now, I've been working with Lloyd on determining this, the value of this Planck biological, we call it biological Planck constant. It's not the same as the physical Planck constant. It's much, much bigger because it involves many, many molecules. And this is just a little bit of math that I don't want to scare anybody, but you can go through the averaging for the metabolic energy stored in the metabolic network. This is the Bose-Einstein formula for averaging, and then we assume the continuous approximation for the frequencies, and because these are waves propagating through the system, and we obtain the through integration we obtain the scaling to regimes actually. At the bottom you see two formulas. P is the metabolic rate or power, energy per time. W is the weight. And in the quantum regime, you have the scaling with the exponent d over d plus 1. What's d? D is the dimension in space. So hypothetically, if you have a highly elongated biological systems, maybe a caterpillar or something like that, d would be 1 in principle. So this would be a 1/2 scaling law, square root. If it's a planar system, let's say a plant which is very surface-like, then you have d equals 2, so it'll be the scaling exponent 2/3, 2 over 2 plus 1, 2/3. And finally, in a fully three-dimensional system like the human being, d would be 3, and there you get w to the power 3 over 3 plus 1, 3/4, the celebrated 3/4 allometric scaling law exponent. So, and then when the other regime, the classical regime, may also occur when you actually flood the system with nutritional molecules, nutrient molecules, glucose, let's say. Then you go into the isometry. So the power, the basic metabolic rate or power P is proportional to body weight. There are some consequences for human physiology, and my collaborator and colleague, Dr. Brian Fertig, wrote a book on metabolism, and he wrote a chapter on this, how this can relate to things like insulin resistance and obesity when you actually have the transition to the classical regime, where your metabolic demands increase linearly with the weight, and that's not good. Okay, so I'll just leave it at this. This is the formula for the quantum metabolism, worth remembering perhaps. So metabolic rate scales with the body weight to the power 3/4 by and large in three-dimensional systems, biological systems composed of enzymatic oscillators. And yeah, there are many consequences, and so that's quite interesting. There are some similarities made between the solids and living systems, but I'll skip that in the interest of time. And here is a slide on the biological Planck constant, which stems from this derivation. It's a lot of calculations on this slide. You don't need to go through it all, but I simply want to mention that when you do the math, it turns out that the biological Planck constant is about 10 to the power 11, larger than the actual Planck constant, which is 10 to the minus 34 times 6.6 times 10 to the minus 34. And this ratio actually makes sense because this would be more or less the number of atoms per mitochondrion in a living cell. That's the order of magnitude. So a single mitochondrion would create some sort of quantized environment, if you will. Okay, so that's on quantum metabolism. Now, what you would normally find in the literature under the term quantum biology is actually photosynthesis as the crowning example of the application of quantum physics to biology. And of course, that's also metabolism, because photosynthesis is the means of energy acquisition by plants and bacteria and some algae. And this story goes back to 2007, where several groups observed the presence of quantum states in photosynthetic systems under artificial conditions, including things called quantum beating, which is a manifestation of a quantum superposition of two states. And that was published in very reputable journals, Nature, Science, and PNAS, and things like that. So it's really very well established. But the mechanism, of course, at the macroscopic level is well known. Photosynthesis utilizes the energy of sunlight. So again, photons in the visible range striking the surface of a leaf, and this energy is used to convert the carbon dioxide and water into glucose. But so this is another example. The molecular basis wasn't really very well understood until 2007, roughly. And this molecular complex is called FMO, Fenner-Matthews-Alsan complex. I'll show you in the next slide. It's also present in these bacterial chlorophylls. And this is it, FMO complex in the C. tepidum. And so that's a very complicated structure, but it was able to be isolated and analyzed in the physical laboratory. Here is the more digestible representation of this very complex FMO. You have here seven molecules forming a quasi-planar organization. They are heterocyclic molecules, and they are all excited by visible light. And these experiments demonstrated, that's even more clear, the experiments demonstrated that the electronic degrees of freedom that get excited by light, so electrons go to a higher energy level and they form a collective wave function. So all seven of these complexes share the same wave function. So that's a truly quantum system where the electronic degree of freedom is delocalized and actually can jump between the different centers, and it has a finite lifetime. So when you excite it, it then comes down to the ground state after a while. The question was, how long does it live? Because that's important. And so there were a lot of papers written on the computational models of this using quantum search algorithms, for example. So the length, so there's this cryptophytes, which also utilize this light harvesting mechanism using quantum coherence. So these are actually very short timescales, picoseconds, perhaps, I don't remember exactly, hundreds of picoseconds, maybe, not quite a nanosecond, but incredibly short, but sufficiently long to enable the energy to be transmitted or funneled into reaction centers where it's utilized for this chemical transformation that is needed for glucose production. So quantum beating and coherence was detected with lasers and so-called two-dimensional spectroscopy. took place with four-wave mixing, and these quantum beats were observed. So these are some slides from the papers that were published, and incredibly precise measurements made. I'll come back to it because one of my colleagues, Professor Greg Scholes at Princeton, was one of the early pioneers in this field. So these effects were observed where superposition states were formed in these FMO systems, and they excited states sample different configurations and eventually direct the energy transfer to the most effective sink, which is utilized for the chemical transformations. So this is a bit, I didn't talk much about it, but quantum computing is another hot topic these days, and quantum computing allows you to search very rapidly a space of possibilities, because the wave function, a bit like a droplet of fluid, spreads out into different possible directions. Unlike sequential classical search algorithms, this is kind of intensely parallel. Okay, so the same way, this mechanism is very fast, as I mentioned, on the picosecond time scale, but is able to search for the most effective energy delivery to the sink. Okay, let me then switch gear for another time. This is now, I mentioned to you microtubules in connection with the cell intelligence, and microtubules are another area where people are looking for quantum effects, and for various reasons. They play very important biological roles in all eukaryotic cells. In dividing cells, they do form mitotic spindles that segregate chromosomes. In as shown here in this cartoon, the microtubules are shown in black, and they are made up of protein called tubulin. So they generate forces in cells. They participate in cell division, transport of cargo via kinesins, dyneins, motor proteins in general, and they also participate in cell motility. But most importantly, in neurons, they form parallel bundles inside axons and dendrites, and that goes to the heart of another sort of far-out theory. Now we are coming to the topic of quantum neuroscience, maybe quantum consciousness. So Roger Penrose, the 2020 Nobel Prize winner in physics, promoted the concept of quantum consciousness in his books going back to the 1990s. And together with Stuart Hameroff, who is an anesthesiologist, wrote many papers suggesting that human consciousness has a substrate based on neuronal microtubules, where quantum effects are involved in our thought processes. And so I will not be able to spend too much time. By the way, Kim, this could be the 5th lecture next year on quantum neuroscience or quantum consciousness.
Yeah.
Yeah, this is now becoming quite a hot topic, by the way. A lot of articles in the popular science sphere and elsewhere talk about quantum consciousness, and that is exactly where microtubules figure prominently. What's interesting is that we talked about photosynthesis as a hallmark of quantum biology, or flagship, so to speak. These 7 complexes I showed you have indole rings, and a similar situation exists in tubulin. I will show that in a minute, but first of all, these are microtubules in neurons. On the right, you see a a kind of a cartoon of the neuron with the nucleus in the middle, and then you have the dendrites on the left and the axon on the right, and these thick lines are microtubules, and the thin ones connecting them are microtubule associated proteins, MAPS, MAP2, MAP-tau, etc. What's empirically known to people in neuroscience is that almost all neurodegenerative diseases, including Alzheimer's and Parkinson's and Lou Gehrig's and even traumatic brain injuries, have one thing in common. There is a problem with the cytoskeleton in neurons, sometimes directly microtubule, sometimes indirectly. So also it is known that during learning, microtubules get reorganized in neurons. So they play some kind of a role, for sure, whether it's quantum computing or classical computing or something else. We are not quite sure yet, but definitely this is important. And this is showing you this network of tryptophans. Tryptophans, by the way, tryptophans, tyrosines, and phenylalanines are the only 3 out of 20 amino acids which can be photoexcited in the ultraviolet range, by the way, not quite visible, but not too far. So they become candidates for something like these photosynthetic processes of energy migration and utilization for various purposes. And this shows you a patch of a number of tubulins forming a microtubule surface, and the red lines show you the potential pathways for these kinds of excitonic quanta that might migrate between tryptophans. So, and that's this analog of what you saw for photosynthetic systems. Remember, there were seven aromatic groups in a FMO patch. Here we have 8 tryptophans, and there are many more tyrosines and phenylalanines I'm not showing. So they can potentially participate in quantum excitations and energy transfer. So we started studying this about 10 years ago with my student, Travis Craddock, who wrote a PhD thesis on aspects of it. And we predicted that there would be maybe even longer-lived quantum excitation than in the photosynthetic systems. And lo and behold, the last 2 years, and this was the culprit, the tryptophan dipole moment of 5.5 dBi interacting with other tryptophans, and we calculated the interactions, and these are the plots of the predicted timescales for the excitations, actually. So it's thousands of femtoseconds, which is basically several picoseconds predicted, on the order of the photosynthetic excitations. So lo and behold, the last 2 years were spent in collaboration with Greg Scholes, I mentioned before, the photosynthesis guru, in his lab. My former student, Arad Kalra, went to Princeton and performed experiments to prove the existence of these quantum excitations. And indeed, we published a paper on this last year, where the results demonstrated clearly that the quantum excitations in tubulin and microtubules exist. And they are actually much longer than these predictions. They can live up to 5 nanoseconds. So we're talking about 2,000 times longer. This is still not quite a physiological scale, because our mental processes occur on the time scale of milliseconds, maybe even hundreds of milliseconds. So we have a big gap, but it's the first step in this direction. The experiments were done under laboratory conditions, not in neurons or in a living system. So that adds to the sort of poverty of the results. But the fact is undeniable that these excitations take place, and they are quite long-lived. And in microtubules, they are longer lived than in tubulin, which would go in the direction of, forming larger structures and creating conditions for quantum excitations to exist. Now, the first question we asked was the time scales, and the second was the range of excitations. In other words, you want something which is large. In the case of photosynthetic systems, remember there was 7 groups connected together was enough for a cell physiological response. And we found that for microtubules, the range, spatial extent of the quantum wave function is about 6.5 nanometers or so, 6, which is the size, roughly the size of a tubulin dimer. So it's not enough to say, oh, the quantum state extends all the way to the entire microtubule and perhaps beyond the neuron is in a quantum state. No, we are not saying that. But again, the conditions under which the experiments were performed were completely artificial laboratory condition of purified protein in solution. There was another experiment under this collaboration with the group of Professor Aristide Dogari from the University of Central Florida. And that experiment actually used visible light. This is called delayed luminescence. And that's actually even bringing us even closer to something physiologically relevant. I want to say the following. The Princeton experiments used the wavelength of about 290 nanometers, which is ultraviolet. And that's a very high energy photon, which would be hard to find in sufficient numbers in the living cell, be it a neuron or any other cell. However, biophotons, as I mentioned to you earlier, can be found in many living systems in the visible range. So this delayed luminescence used light in the range of 630, I believe, nanometers. So that's red, I think. Anyway, it's visible, and it involved lasers shining photons on the sample of tubulin or microtubules for about 2 minutes, and then observing the decay in the intensity of the re-emitted light from the sample, like so. And the question was, how long does this take to re-emit the photons? And you would imagine, if you shine light, let's say on a piece of material, some solid or a desk or plastic, the light is absorbed and some of it is reflected, but it's super fast. You don't see any delay in emitting light. There are some chemically engineered materials which demonstrate delayed luminescence, which is re-emission of light over, let's say, seconds, even sometimes hours. But this is really different because we are talking about tubulin microtubules in these experiments in the solution. So okay, here it is. These are the results of these experiments, and you can see here several situations where you have tubulin, unpolymerized tubulin in the sample exposed to this light, and we used colchicine to prevent polymerization. Then we have microtubules stabilized by Taxol, another chemical agent, and then we have a coexisting situation. So in the upper right corner, you see the curves showing you the delayed luminescence, the intensity of light as a function of time. And it amazingly, it takes minutes to re-emit these photons. So so that's really not only a physiological timescale, but well within it. And what does it tell us? That it tells us that, oh, and one more thing, the intensity, both the intensity and the duration, the lifetime of the re-emitted light is longer for microtubules than for tubulin. So what does it tell us? It tells us that that it is quite possible to have interactions between microtubules, for example, in neurons, and photons generated internally or externally, whereby these photons are absorbed for a while and perhaps interact inside microtubules, generate some structural changes, and then they get re-emitted at a slightly different wavelength and with a substantial delay in time. So that would be going very much in the direction of biophotons being utilized by structures such as microtubules and potentially quantum processes such as memory encoding or some kind of computation. Let's put it this way. Now, so this is the question that would naturally occur is where do the photons come from? And I mentioned earlier that mitochondria emit photons as a byproduct of their activity. And by the way, I didn't mention, but it is well known to cell biologists that mitochondria actually are closely associated with microtubules, and they indeed move on microtubules being carried by motor proteins. So there is a very close proximity to the source of photons, mitochondria, and microtubules, which could utilize these photons for the processes that we suspect have something to do with cognitive functioning. So these are the final slides I want to close the lecture with, asking some questions and maybe providing some potential answers. So now we're going to the toward the neuroscience end of things, of quantum biology. The first question is, what is consciousness? And second is, where is memory stored in the brain? At the molecular level, we know that there are some areas of the brain responsible for different functions, including memory, and sometimes when people have brain damage, other areas take over. But we're not talking here about macroscopic dimensions of where potential memories are stored, but microscopic mechanisms. Then there are some issues with the computational power of the brain. You know, how does it compare to what we know from computer science and electronics? And then, of course, is information processing in the brain classical, quantum, or some other type, maybe fractal, maybe resonant? So these are the big questions that we've been trying to address. And I kind of started from microtubules. I will come back and end with microtubules because there is potentially a lot of functional roles microtubules can play. So I showed this statement made several years ago by Robert Epstein. Your brain is not a computer and doesn't process information, retrieve knowledge, or store memories. In short, your brain is not a computer, which I completely disagree with. And of course, there are some ridiculous statements like, where do you find a copy of Beethoven's Fifth Symphony in the brain? This is not serious, but actually, we know that we do perform computing. We can computations. We can perform complicated operations. We can also I think much of the brain's activities is algorithmic, but not all. And Roger Penrose, in his very elaborate books, made a very strong case that there are numerous examples of non-algorithmic behavior of the human brain. In some cases of chess games or intuitive solutions to problems that the computer cannot solve. So I think the answer is somewhere in between. The human brain is to some degree a computational organ, a device, performs computing, but it's also more than that. Not only, just like the human body is a machine, but not just a machine. So I'll start with the sort of very, very general descriptions. We don't have to go into the details of the types of brain cells. We know that neurons are responsible for signaling and signal processing, and there's about 100 billion neurons in the brain, 93 billion, and to be specific, I think this number has been debated in the literature, but let's say 100 billion neurons, and on the average, each neuron has 10,000 synaptic connections. Some neurons can have up to 100,000 communicating with that many other neurons, so that's mind-boggling. So we have about 10 to the 15th synapses, which operate at about 100 millisecond time scale. So in principle, we can have up to 10 to the 16th synaptic operations per second, which, by the way, overshadows what some of the most powerful supercomputers can accomplish these days. The Blue Gene, IBM's Blue Gene performs 10 to the 15th operations per second. But, sorry, what really is, I don't have this number here. What was more remarkable than the sheer number of, this here, the sheer number of computations per second is the energy expenditure. So the IBM's famed Blue Gene supercomputer consumes 1.5 megawatts, million watts of power, joules per second, whereas the total energy consumption of the brain is 25 watts. And that accounts for everything, including temperature regulation, including protein synthesis, repair, material transport, etc. So my kind of conservative estimate is that the computational part, the cognitive cost, is about, let's say, 5 watts. What is 5 watts? Well, it's about the amount of power that a pocket flashlight uses. That's what we use for, let's say, all we do now, listening. Some of you are maybe writing something on the side and thinking about some other things, and twitching and looking at your computer. And so all of it is only consuming a minuscule amount of energy, orders of magnitude, let's say 6 orders of magnitude or so, 1 million times more efficiently working than a supercomputer. So how does it achieve it? And I think one, of course, glib answer would be, well, it's a quantum computer, stupid. So it's very efficient. But of course, we have no proof that it's a quantum computer, except for what I told you just a few minutes ago, that if it uses microtubules, and microtubules use biophotons, and that is done by quantum energy transfer, then it could be incredibly efficient. And of course, we are at the top of the evolutionary tree. So after 4 billion years of evolution or so, Mother Nature has created a quantum supercomputer that is called the brain. Okay, so that's now let's be serious for a second. So the question is the brain a quantum computer? Is there a potential collapse of the wave function? And actually, Hameroff and Penrose claim that when we realize a thought that, you know, before we realize a particular thought, we may have several ideas at the same time, like a quantum superposition of states. But when we actually realize the thought, let's say, you know, I really want to have Coke, not Sprite today, to drink now after my talk, or I'd like to have coffee, not tea. This is like a quantum wave function collapse. So before we realize things, before we process things, we have superpositions of quantum wave functions. When we do commit to something or decide on something, that's the collapse of the wave function occurring. Now, if this is true, then there are some very interesting questions, follow-up questions. Number one, with the Heisenberg uncertainty principle at work, then, because it's a quantum state, we have built-in uncertainties. There may be some limitations on predictability of the human brain, which I wouldn't mind at all. I think it's quite conceivable that our brains are impenetrable or not completely predictable in their functioning. The second, of course, is implications for artificial intelligence, which is all the rage these days, and everybody thinks that solves all the human problems, that we can use artificial intelligence to replace creativity, insight, intuition, or sheer power of the mind. I tend to believe that artificial intelligence is just that. It is artificial. It's not really intelligence. It has nothing to do with knowledge, and it has limitations, which, well, perhaps down the road there'll be quantum artificial intelligence developed. I don't know, but at the moment, if we accept the brain's potential for quantum computing, it's some, not everything, but some of the processes, then of course we have a reason to believe that our brains cannot be easily replaced by computers. Okay, so this is roughly speaking a cartoon that represents the current interpretation of the Penrose-Hameroff theory of consciousness, where you have the neuron connected to another neuron through a synapse, and inside this neuron you have bundles of microtubules represented in blue, with the tubulin dimers, the functional units and structural units of microtubules shown in red and then magnified. And you see here this protein qubit where microtubule could be in two quantum states, maybe simultaneously, and then the state could collapse onto a single state. So that's in a cartoon representation, the Hameroff-Penrose orchestrated objective reduction model, which predicts the fundamental role of tubulin and microtubules in quantum processing at the subneuronal level, and somehow, without really going into detail or specific explanation, if there was a quantum state over an entire microtubule, perhaps the whole neuron would be in a quantum state, and then you can imagine neurons interacting with other neurons in a quantum-like fashion. And by the way, there are some other candidates for quantum states in cells, and one of which neurotransmitter release to trigger synaptic activation was touted to be a quantum tunneling phenomenon by none other than Sir John Eccles, a Nobel Prize winner in medicine and physiology. He published a few papers in the 1990s with Beck, claiming that neurotransmitter releases a quantum phenomena. There are also, I mentioned Frolic, so there could be quantum states in the membranes of neurons, and now we have microtubules strongly favored by Penrose and Hameroff. Now, we know a lot about microtubules, and here we kind of demonstrate this hierarchical organization. On the left, you see the axon and the synapse, and then you have the in the middle, next to the right, a bundle of microtubules interconnected by maps. Then you see a single microtubule, and then you see in the building block alpha beta tubulin dimer, where apparently all the action is in terms of quantum degrees of freedom. Microtubules are not isolated. I told you they interact with other microtubules by microtubule-associated proteins. They also interact with voltage-gated ion channels. There could be a lot of potential for interesting interplay. And these are some other representations of these structures, microtubules. We now have atomic resolution models of the crystal structure of tubulin, and a lot has been done on understanding the interactions of tubulin with other structures, including, and I'll maybe spend a minute or two on this because it's important in connection with consciousness. So tubulin interacts with many drugs, some of which are chemotherapy drugs used for the treatment of cancer, such as Taxol or zinc alkaloids, but also interacts with anesthetic molecules such as isoflurane, desflurane, halothane. All of these interact with tubulin, which would indicate that there is a possibility of explaining anesthesia, one of the miracles of medicine, still unexplained completely. That would give us a potential explanation of how anesthesia works if these molecules, anesthetic molecules, interact with tubulin, and tubulin is the place where quantum consciousness occurs, then anesthetics could destroy this quantum coherent state by binding and interfering with the interactions. So in these experiments I mentioned to you that took place at Princeton and Florida over the past 2 years, we also did the measurements on the lifetimes of quantum states of tubulin microtubules in the absence versus the presence of anesthetics. We used 2 or 3, I don't remember now, different anesthetic molecules. In all cases, anesthetics shortened the lifetime of these quantum excitations, not by much, by about up to 20-25%. So, but measurably and reproducibly. So that was an encouraging result. What was slightly discouraging was that we also used a negative control, picrotoxin, which is not an anesthetic molecule, and binds to tubulin and had the same effect. That doesn't invalidate the result with anesthetics, but still, we still have some way to go. Okay, so this is how microtubules are made. Now, I mentioned to you I will finish in a minute, so maybe there are some questions, but I want to say the following. If microtubules are involved in computation at the neuronal level, then the potential for massive amounts of information storage and information processing is incredible, because they have several degrees of freedom, each of which can be utilized as a channel of separate information storage and processing. This includes so-called C-termini, which are protruding short peptides protruding from the surface of tubulin. So there are at least 4 states, one bound to the surface, one unbound. So 4 states per tubulin dimer. Then there is the possibility of electron hopping between 2 monomers in the dimer. Then we know that there are conformational changes related to GTP state. So GTP binds to beta tubulin and can hydrolyze to GDP. So there is 2 states per dimer. And there could be also additional states related to, which I mentioned, tryptophan excitations, as well as phosphorylation states. So serines in C-terminal can get phosphorylated. Well, they do, we know that. And so when you add it all up, you can have up to 32 states per dimer, and there's about 100,000 dimers in the neuron. So when you do the calculation, you can obtain about 100 kilobytes of memory storage per single microtubule, if all of these states were utilized, or 1 gigabyte per neuron. And with 100 billion neurons in the brain, and maximum utilization of all of them, we could have 10 to the 20th bits per brain. This is an unimaginable amount of information, and I did the calculation. This would easily store the memory of the entire lifetime if somehow there was a mechanism for encoding and decoding information from neuronal microtubules in the brain. We could do that for memory storage and retrieval. Now, whether it's true or not, time will tell, but the potential exists. Okay, so now this is a bit technical. So the next question is, the first is the storage, how much information, how many bits of information can be stored in the brain, and the second, how much can be processed, and that means bits per second. So that depends on the speed of operations, and then we know that our overall processing time is about 100 to 500 milliseconds. So that would increase the number of processing operations by another factor of 10 or so. But then there are some other issues such as would this be too expensive energetically? Of course, we don't have to use all the brain all the time, all the potential of the brain all the time. And I think Einstein said that we use about 10% at most. He did. So anyway, let me just end on something which is nice and visual. Oh yeah. So by the way, yeah, maybe I'll stop here. I mentioned that if we used all the microtubules and all the neurons in the entire brain, we could encode about 10 to the 20th bits of information. In fact, the father of computer science, John von Neumann, in 1950, made a bold prediction that the human memory can consist of 3 times 10 to the 20th bits, which is even higher than my estimate. But yeah, so that's much better than some of the other estimates. People were Thomas Landauer, in his 1986 paper entitled "How Much Do People Remember?" Very pessimistically estimated 10 to the 9th bits of information stored. I think our human lifetime is about 2.5 to 3 billion seconds. So that would mean that we only record, according to Landauer, not me, record 1 bit of information every 3 seconds. Anyway, so I'll leave you at this. This topic is never ending, and maybe next year we'll spend a whole hour on quantum brain, and hopefully there were some interesting and intriguing concepts that you didn't know existed and would like to delve deeper into. So there's a lot of literature, and if you want any pointers in the direction of current literature, I'll be happy to answer your emails with questions. Thank you.
Okay, maybe you could unshare and we can
Yeah.
discuss. Yeah. So, just before this, Sherry Heel and I did an interview, and I mentioned that in the course we have international peer review and sort of expert review. And many years ago, when you had just one lecture, the peer reviewer said that one lecture was incredibly dense and we needed to have more. So I'm quite in favor of going from 4 to 5, and we'll see what happens. We could always shrink back to 4 after that
Yes.
if we need to.
Yes. And
But
of
it's
course,
worth a try. Yeah.
of course, the students are the ultimate arbiters of
Yes.
this.
Yes, indeed.
By the way, no, we can stick to 4 or go to 5, but I think what I would like to know is what the students feel is interesting, worth keeping, and what is maybe not completely properly described or would like to hear more about. That would give me some direction, because obviously I'm too close to the topic to
Yeah.
see.
One simple metric here is most other things we teach in this course you can find in other courses, but most futurism courses do not have quantum biology at all. They seem to be afraid of it. They're frightened of it. So I'm very proud that we have something that those other courses lack, and hope that the students share my enthusiasm
Yes.
for
And
the
in the fall we'll
uniqueness.
have the quantum medicine book that will be published by then, I think. So
Yeah.
I hope I'll be able to share at least a PDF copy with the students.
When the course began, in the initial focus groups, we talked about my writing a book concurrent with teaching the course and the students taking part in this. So it would be nice if something at least mildly like that happened next term, with your book and the students getting, some sort of personal knowledge of what the writing of the book is like. And you can argue that this course played no role whatsoever in the book because you could have done the book without the course. But on the other hand,
Not really.
your confidence that there's an audience out there a lot greater because of the course, because there are thousands of people watching your videos, and it makes it seem like, that there's the potential.
Well, it did play an indirect role, because through this online video, I was contacted by a doctor from New Jersey with whom I then worked for several years on his book. And then we decided to put together our book on quantum medicine, because he's a medical doctor, I am a quantum physicist, biophysicist. So kind of a combination occurred. And yeah, so the first trigger for it was the video that you produced.
Yeah. Well, no, I'm happy to have been a force for good in this instance. Okay, I've done enough talking. We need to hear from a student or 2 students or 3 students. Yeah, we've got 6 minutes. Sheraheel?
Yeah, I'll be honest. Some of the stuff we discussed in these last 4 lectures has gone straight over my head. I haven't quite grasped it, but what I can grasp is sort of the intersection between not just quantum biology, which we talked about, but also a lot of quantum physics and quantum chemistry, if those are the correct terms, but a lot of the intersection between the sciences, and translating it to sort of the focus of our course, on technology and the future of medicine. So by and large, it was a very interesting series of lectures, and especially sort of parts of it that I'd learned in previous courses or similar introductory courses in university, and then sort of building on that. Yeah, I guess some of it, I suppose, was maybe a bit too advanced for me, or perhaps I just
So
didn't have enough time to
you're in
process it.
your
But
first year, right?
yes, and
Yeah,
that
so
might have something to
you're
do with it.
amongst the youngest students we've ever had. And I think that's kind of
Well, you
cool that I was interviewed today by a very young student, you know. It isn't that you needed a lot of seasoning to do it. You could do it
Well,
without
and
seasoning.
that's sort of, I guess, the other thing is with what I'm saying is, I guess my experience has been limited in terms of other knowledge in classes in university, so that could be part of why perhaps I didn't understand some of it, but certainly the
Well,
important aspects were definitely conveyed.
you should be proud of yourself, because I don't think this course could be given in these lectures could be given any other course. If I were to give it in, let's say, for my even graduate students in physics, they would probably chase me out of the classroom.
Yeah, well, there's some history there, isn't there, Jack? So in the beginning, when Jack and I didn't know each other very well, he had a standard lecture on computational drug design, and he decided to give that instead of quantum biology.
Because I was lazy.
I'm a pretty jovial guy, but I was not happy, not a happy camper.
I know.
So he sort of learned that I'm not looking for conventional content in this course. Yeah.
Yes, I remember that vividly. But you know, this series of lectures was not supposed to give you any concrete knowledge you can just use tomorrow. More to inspire you to look for things which are not even completely understood by the most advanced scientists, you know. Yeah, I think this is a transition from undergraduate education to graduate, where you discover things that are, you know, in textbooks, but in reality, it's not quite so.
Yeah.
So
Zayed, Scott, any thoughts at this point? And do you think future students should have more of this or less or none, or where do you stand on the 5 lectures versus 4 versus 3, 2, 1, 0? Yeah, what's your preferred number?
I think the more the better, just because
Okay.
as a topic, I think it's very either new or foreign to students, because unless you're like actively studying quantum biology, quantum mechanics, whatever it may be, this is a very tough topic to wrap your head around without the prerequisite work. So the more lectures that could be made available to kind of conceptualize it, the better, I would say. But I kind of appreciated it. So when we like entered the course and when we go about our lectures, like we think about futuristic ideas and we think about AI and we think about how that affects medicine. But then sometimes it's good to take it back a notch and look a little bit deeper under the microscope and things that can't be seen by the naked eye because they're also just as relevant. And so that's where I found like the value in learning about things at the quantum level. So yeah, I think it's a great addition to the course. It like provides you a good perspective from, you know, very macro scale things to then very like micro, even smaller than micro, obviously. So yeah.
Scott, you were the source of the idea of getting a PDF of all the slides.
So he's in a quiet
what's-
library, right? Okay, can't talk.
Oh, I see. Okay. Okay.
But
Yeah.
that's, thank you very much for your feedback and for the time you took to listen to these seminars or lectures. I will, we kind of try to improve every year. Maybe next year
Yeah.
will be still better, but
Yeah. Onward and upward. Well, yeah, this is a great final lecture, Jack, and thank you very much for that. And we'll look forward to even greater quantum biology accomplishments in the course in future years. And it's nice to have these recorded comments, so future students won't say, well, did any student ask for this, or did you just arbitrarily give it to us without testing the idea at all? So yeah, we tested it with actual human students. Yeah, that idea. Okay, well, that's great. So I'll see you guys and the rest of the students face to face on Thursday at Echo 1432. It's kind of a famous room. You probably got the same email I did. They're trying to popularize Monday Study Hall, and that's the room where the Monday Study Hall has been. I don't know whether that's anything important. Okay, so Jack, thank you very much. We look forward to next term and the terms after that. And yeah, for the students, I'll see you face to face on Thursday.
Bye-bye.
Okay, take care. Bye-bye.